Imagine a baby whose world begins in shadows, struggling to track faces, respond to light, or recognize movement. For families affected by Leber Congenital Amaurosis Type 10, this early vision loss begins in the very first months of life. And at the center of it all is a single gene, CEP29. This gene carries a tiny but devastating defect that prevents the retina's light-sensing cells from working properly. For decades, doctors could only watch as vision deteriorated. But then something extraordinary happened. Science decided not just to treat the symptoms, but to rewrite the code at the heart of the disease. The first breakthrough came with CRISPR gene editing, a technology that works like microscopic scissors. During a precise retinal injection, CRISPR enters the cells and cuts out the defective part of the CEP29 gene. The goal is simple: allow the cell to produce the healthy protein it has been missing. This is the first time gene editing has ever been tested directly inside the human body. Early results show signs of improved function, a hint that damaged cells may be revived. But the innovation does not stop there. A second therapy, an RNA antisense treatment from ProQR, approaches the same problem differently. Instead of cutting DNA, it adjusts the genetic message inside the cell, allowing it to produce the protein needed for vision. Early patients have shown meaningful improvements in visual acuity and retinal structure. Two different technologies. One goal: restore light to a condition once believed irreversible. These treatments are still in clinical trials, and much work remains. But for the first time, children born with LCA10 may have a chance to maintain, or even regain, precious vision. This is not just progress. It is the birth of a new era. An era where inherited blindness can be challenged at the level of DNA itself. And for families touched by CEP29 mutations, this marks a future filled with possibility, courage, and hope.